We begin with the topic mRNA vaccines and therapeutics manufacturing, and welcome Dr. David Loong, Senior Consultant, Novel Modalities Asia Pacific Bioprocessing Strategy from Merck Life Science. Welcome, David.
Hi, everyone. Just getting used to this interface. Hi, thank you for the kind introduction, Linda. I just have 2 slides to talk about mRNA manufacturing and vaccines. I will be giving a presentation later this afternoon. So that contains more of the context and more detail into the bioprocessing aspects. So really the 2 slides that I have are really just as a very quick overview, and then the key points for the scaling up of our mRNA vaccines and therapeutics. So if we go to the first slide. So the rapid development and deployment of mRNA from bench to process scale to make the COVID-19 vaccines has meant that there hasn't been time to develop in industrial or manufacturing-ready processes. So many of us may be familiar with the common lab-scale processes of isolating mRNA, maybe when you were trying to isolate RNA or was trying to sequence some DNA using a commonly used kit as the QIAGEN kit. You do a lot of shaking and spinning down and use some solid phase extraction, etc. And generally, you would use solvent extraction, precipitation. We've probably very familiar with using those buffer mixes that come in those kits, and then you precipitate out your nucleic acid and then do some purification and then concentrate again purify again by concentration. So those unit operations with solvent extraction, precipitation, and are not suitable for when you want to go into a bioprocessing scale or manufacturing scale. So these need to be replaced by scalable unit operations such as chromatography or ultrafiltration and diafiltration. So those are the common challenges for scaling up from bench to manufacturing scale. And then the next slide, please. Zoom back out again. All right, and then the next key feature for mRNA manufacturing is the formulation of the mRNA into the nanoparticle. So this has really been key for the success of mRNA therapeutics. RNA therapies have been around for a very long time, but they just haven't seemed to have been able to work. And it is really the formulation, right now, the lipid nanoparticle formulations that have made them be successful and work. So this is key. And there's a whole speciality and expertise in this area, which we're all trying to get to grips with and understand, what makes a successful formulation, what is needed, because there is no one-size-fits-all formulation. There is a formulation development that's needed. Moderna invested as much in their lipid discovery as some people would do in drug discovery in terms of synthesizing novel lipids, changing the organic chemistry and the structure of these novel lipids around. So yeah, this is where the magic, as it were, that makes mRNA work happens. So yeah, those are just the 2 quick overview slides that I have. There will be a lot more in the presentation later on. So yes, very happy to take questions. I think the last slide is just oh yeah, there we go. Okay. So, so we have some questions here, and I will take the first question, which is in terms of facility design, how complex is a mRNA facility? So the interesting thing about mRNA manufacturing is it doesn't have a living component to it. So unlike traditional biopharma processes, you don't have like a cell line, something that's living, you need to feed it. It's enzymatic process for mRNA. And this means that generally all the unit operations from the plasmid template onwards can be conducted in one space. You might choose to segregate them depending on the closeness to to the drug product. Maybe you want to have a higher grade suite for your final formulation and your fill finish suite. But the other unit operations, the in vitro transcription reactions, the purification reactions, those can all be done in one space. And I will take the last question here. So this is here, any concern about contamination by adventitious agents? So these are the same. So these adventitious agent contamination is the same for all bioprocessing. You can think of mRNA as being processing as being a extended downstream processing of pharma, because most of the unit operations will be in mixers or in chromatography or TFF, filtration. And so right now, there is no defined regulatory requirement for other than the usual guidelines for sterility testing for standard biopharma. There is no special guideline for mRNA for adventitious contaminant. For product impurity contaminants, there's increasingly detailed guidelines that are coming from the agencies. But right now, it would just be your standard biosafety contamination guidelines. I think one thing that can be recommended is bioburden reduction filtration at as many points in the production process as possible. Moving adventitious agents, the mRNA itself can be filtered depending on the membrane cutoff. It's not like the size of a cell, so or the size of a lentiviral particle, which has issues with 0.22 micromembrane filtration. So that kind of strategy for control would be adequate for the mRNA process. Okay, so we have one more question here. So how can we assure that processes, devices, and reagents are RNAs? So this would be RNAs free. So this is the biggest challenges. How do you, RNases are ubiquitous in the air. They degrade RNA, and it will be challenging to have a whole facility RNAs free, to have your entire water supply RNAs free. So what is possible? It is a strategy that maybe you just want to have your RNAs free water at the final filtration step, or at the closer to the end when you have a more precious RNA product. And then for earlier stages in the process, you do a sort of process risk assessment and process modeling. Maybe you do test your WFI for RNAs, but you don't necessarily need to ensure it is RNAs free. But knowing your level of RNAs, then you use that WFI water in your process and see what yield you get. Maybe you might do some development to say that let's try it with RNAs free water. Is the yield much improved? And if it's not, then maybe we can tolerate using WFI water. Generally for devices like filters, chromatography, you would flush with WFI water, or if it's a very crucial part and you feel that RNAs is degrading your product, then you would flush with RNAs free water to ensure that unit is clear of RNAs. So the next question I will take is how are measures used to ensure the stability of the lipid nanoparticle? So can we improve the stability for it to be stored at 4 degrees? So yes, indeed, a lipid nanoparticle with mRNA is stable at 4 degrees. This is the CureVac lipid nanoparticle. And this is because they have proprietary lipids and they have a proprietary formulation process. So it's really the interaction of the RNA, the various lipids. There's currently, people talk about 4 different types of lipids that add different structural components and structural stability. So it's really the electrostatic, the physical chemical interactions. If we go back to our chemistry, the van der Waals interactions, the hydrophobic, hydrophilic interactions within that little lipid nanoparticle that hold it together. So optimizing all those molecular interactions, as well as how they interact with the RNA particle itself, that is key to ensuring the stability of the lipid nanoparticle. And if you are, I guess, if you do enough development, then you can get to that stage to avoid cold chain supply. And the reason we had such cold chain supply is because there was such urgency to develop there was time to spend on the development of more stable particles. So our question was, what is the biggest advantage of mRNA vaccines over traditional vaccines? So what we've seen, the biggest advantage is speed of development. I mean, who would have thought we would have had a vaccine in 1 year? Everyone was saying it's going to be at least 2 years, at least 4 years, but 1 year we had a vaccine that works. And this is probably the big. Another advantage over the traditional vaccine forms, this is where we're talking about live, attenuated, and inactivated vaccines, is that you don't have to handle the actual infectious organism to make the vaccine. You just from genetic information alone, you can isolate and work with what is the antigenic part of the vaccine without the handling the dangerous part of the vaccine. I would take the next question, which is what are the major constraints during mRNA production? Okay, it's jumping around a bit. It's all right. So I think for mRNA production, right now, one major constraint is that there is not much known about existing processes, and there's so many different types of processes that are out there. There is no there's no industrial template, put it that way. For example, everyone knows for a MAP process, you use a CHO cell line. You probably have a 3-step purification. You do this final filtration. And the ecosystem around the MAP process. Whereas for mRNA, the commercial processes that are out there are very different from one another. Some use chromatography, some don't. Some are very large scale, some are very small scale. So this is I would say is a major challenge, is that we don't have something that is standardized. And because there's no standardized template, it means then supplying a solution is difficult, because we have to really go back to basics and back to our engineering and physics to think about what might work, what might not work, what are the risks and watch-outs, and then do the risk assessment. So all these risk assessments that need to be done, which are accepted as part of a course for established biotherapeutics. Okay, the next question is, so mRNA is a novel modality. Functionally, do we consider it more as a chemically synthesized therapeutic or a biologic? So this is very interesting. I consider it's an enzymatically synthesized therapeutic. So there is no live cell upstream. There's, as I mentioned before, there's no E. coli, there's no CHO, there's no HEC. There's nothing living in the manufacturing process. So this means that the level of biosafety and the level of, I guess, the fragility of the process is a lot more reduced. You don't have to worry about handling upsetting or causing a cell to die or not to produce. So in terms of the function, it has the same function as any biologic. It reacts on a biological target. It induces an immune reaction. It reacts on the immune system. So in that sense, it works functionally as a biologic, but in a processing sense, it's more similar to a synthesized product. In fact, small interfering RNAs, these are RNAs that are less than about maybe 150 to less than 150 nucleotide bases. They're actually chemically synthesized. mRNAs, which are around 2,000 to 3,000 nucleobases, because of the length, are enzymatically synthesized. So the next question, we're at time. So I'm happy to take more questions after my presentation. Thank you very much for the interactions. I look forward to speaking to you in a couple of minutes.